Thank you for watching the video. My name is Fanying from Genomic Research Center at AbbVie. Today, I would like to talk about our project in which we identify disease-relevant cell types in atopic dermatitis by integration of GWAS signals with epigenetic and single-cell RNA-seq annotations. Here is my disclosure and acknowledgement. This is an overview. The objective of our project is to identify the disease-relevant cell types in atopic dermatitis by integrating GWAS signals with epigenetic or single-cell transcriptomic annotations. We are interested in the question because the causal cell type in many autoimmune diseases, including AD, are still unclear. The answering the question is critical for the choice of a proper experimental model system for functional validation, can help us understand the disease etiology and also facilitate the development of targeted therapies such as antibody-drug conjugates. As for the conclusions, when we use tissue-specific or cell type-specific epigenetic data as annotations, we found that variants associated with atopic dermatitis are mainly enriched with T cells, including Th2 cells. With the single-cell RNA-seq data of skin tissues from healthy donors and atopic dermatitis, we constructed gene programs, including cell type programs and disease progression programs. For cell type programs, we found an enrichment of T cells, NK cells, innate lymphoid cells, dendritic cells, and Langerhans cells. As for disease progression program, we found an additional enrichment of keratinocytes. So to prioritize disease-relevant cell types, the general strategy we're using is SNP enrichment analysis, which integrates the association signals from GWAS of the disease we're interested in with functional annotations that is cell type specific or tissue type specific. Generally speaking, we are overlapping the SNPs from GWAS with these cell type specific regulatory regions. If this overlap occurs more frequently than expected by chance, we assign a higher enrichment score to the corresponding cell type, and this is more likely to be the causal cell type. So when we have the epigenetic annotation to integrate GWAS, we use a method called LDSC, developed by Elkis Price Lab at Harvard University. Here is the overall information of the GWAS. It's the meta-analysis result of UK Biobank and EAGLE for atopic dermatitis. Together, 38 independent signals across 21 GWAS risk loci were identified. As for epigenetic annotations, we tried both roadmap data and also attack-seq data isolated from peripheral blood from healthy donors. And on the left is the roadmap results. Each dot is a sample, and the different colors corresponds to different tissues. As you can see, the only one that are considered significant all come from blood. To figure out what cell types in the blood are enriched, we went to the attack-seq data of peripheral blood, which are either stimulated or under resting state. And as you can see, majority of the cell types that are enriched with LDSC are all different subtypes of T cells. We also observed one subtype of NK cells. As a positive control, it's good to see that there is an enrichment of Th2 cells, which corresponds with the fact that atopic dermatitis is considered an autoimmune disease driven by Th2 cells. When we come to single-cell RNA-seq data, we constructed gene programs to identify relevant cell types during disease initiation. We construct a cell type program. These are genes that are specifically expressed in one cell type compared to all the other cell types in the healthy tissue. And to identify relevant cell type during disease progression, we construct a disease progression program. This time we compared the differential expression between cells of the same type in disease tissue relevant to the healthy tissue. So this is the method we developed called MAGMA-SC to conduct enrichment analysis with the single cell annotations. The method is built on an existing tool called MAGMA. Here are the citations. And we also borrowed a lot of ideas from the bioRxiv paper here. This method consists of the two steps. The first step is gene level analysis, in which we conduct an association test between the phenotype, which is cases or control, against the principal component matrix for each gene. In the association test, we get a p-value, which was transformed into z-value by per bit function. In the next step, we conduct a gene set analysis to test whether the genes in a gene set are more associated with the phenotype of interest than other genes. This gene set is quite flexible. It could be gene in a pathway such as the ones in gene ontology analysis, or could be cell type specific markers. In our case, they come from the cell type programs and disease progression programs in the previous step. As for the formula, on the left is a vector of n. Usually these are the values we got from step 1 for all detectable protein coding genes. And then on the left, the key component is the indicative variable. In traditional MAGMA, they consist of binary values, with 1 indicating a gene is in a gene set, 0 indicating the gene is not. But with MAGMA-SC, instead of a binary value, we make them into continuous values ranges between 0 and 1. These continuous values are generated by minimal maximal normalization of the p-value from Wilcoxon test when we constructed the gene programs in the previous slide. The advantage is we utilize all the detectable protein coding genes, and we don't need to arbitrarily set a threshold. To conduct a regression test, we can estimate the effect size beta. If it's more than 0, it indicates there is a positive enrichment, and we also have a corresponding p-value. And here is the final results. The single cell RNA-seq data set we used is from human cell atlas, human skin. Here's the number of healthy cells and cells isolated from atopic dermatitis lesions. In this analysis, 42 clusters were identified by the authors. We also manually clubbed them into 14 broader categories of cell types. So the first column of the figures corresponds to cell type program in healthy donors. The second one are the cell type programs in atopic dermatitis lesion. And the last one is disease progression program in atopic dermatitis lesion relative to healthy controls. So to highlight some results, as a positive control, we observe there's an enrichment of T cells in all programs, and also innate lymphoid cells or NK cells. These results are also reproducible when we conduct analysis with the epigenetic data in previous step. Also, in this paper, they highlighted a novel subtype of cytotoxic T cell with a high expression of IL-13 and IL-22. This is a novel cell type that is specific in atopic dermatitis skin sample. And it's good to see that this is also the cell type with the highest enrichment signal. Another cell type I would like to emphasize is the Langerhans cells. This is a cell type specific in skin tissue sample, which has features of both macrophages and dendritic cells. The last cell type I would like to emphasize is keratinocytes. This is the most abundant cell type in skin tissues. A damaged epidermal barrier with activated keratinocytes is a hallmark in atopic dermatitis. the activated cells release pro-inflammatory cytokines, chemokines, Th2 promoting cytokines, and contributes to the pathogenesis of atopic dermatitis. We didn't find the enrichment of keratinocytes in any of the cell type program or in epigenetic data when we use keratinocytes from normal sample from roadmap data. Instead, we only found enrichment in disease progression program. The results indicates keratinocytes may not be the causal cell type during disease initiation, but it could be a relevant cell type during disease progression. That's all I would like to present. Thank you.